All right, folks. So let's start. Just to show you that where you will find today's material, uh, because I thought that last time somebody told me that they didn't find that where the last slides were posted, and I was kind of shocked. You tell me that after the exam. I mean, like, literally, I was like, anyway, so I just don't want to get into that. But see, that exam 3 review material, here you have exam 3 review PowerPoints, all the 4 topics with those star mark slides. Then you have the recording, and the corrected slide recording starts at 28:41. You just watch it just as it is. There's no problem with that. Here is the Module 3 Honor Special Topic Review PowerPoint that we are doing today. And I thought it's like those star mark slide with the questions, so I will do it, but I appreciate that you guys presented it last time. But I'm going to present the whole thing. That will be a little bit probably streamlined, a little bit, not necessarily better. But let's take a look at that. I do not need Module 2. So right here is your Module 3, and you have it in both places. See, coronavirus, SARS-CoV-2, COVID-19 related topic related for exam. Here you have the PowerPoint. So this PowerPoint and the exam review PowerPoint right here, they are both the same. So I will just write right here, also in the exam review module, you will have the recording link right here, as well as you will have the recording link right here so that you don't miss this last one by any means. All right. So the second thing is that some of you already asking is that exam is Thursday or Thursday to Sunday. I just wanted to hold on because as soon as I say that, okay, all right, it's up to Sunday, then I was like, I'm not studying. Okay, all right, done. I'm going to study only on Friday and Saturday, and that's it. No, I mean, like, I'm not saying you guys. I mean, like, you guys are coming to the class every day and probably way more serious than many of guys. But again, that's why I just wanted to hold. So it will be open until Sunday, 11:59 p.m. That's just how it is. But just to make sure that that will not be the case for your final. Just I wanted to make sure. I will open the test early. So say Wednesday, I will open the test, or that's fine by me. But you have to finish that by midnight. This is the rule set by the university. So that's basically, I have to follow that. I have to, well, university set the rules, like the time, 7 p.m. to whatever it is, but I will extend it to up to the midnight. Yes?
It's still going to be online, right? So
it's exactly the same thing. You can take it from home, wherever you are. Yes, yeah, absolutely. That's what it is. And so that's pretty much what it is. And you know that where things are. And so let's start with the material. So again, this will be the last slide. This is just small talk for the honor students. I send a pretty harsh email to the honor students. And again, that one email, every point is not directed to everyone. It is collective things, so do not feel bad about it and do not get scared. You're still doing a great job, many of you. Now here, what you are going to see, that some of the slides you are going to see extensive highlights and the star marks. And those are extra important for your exam. We're going to talk about that. Does that mean the other slides, which doesn't have a star marks, are not at par? No, that's not the case. The way I chose that there are some topics which is a little bit more difficult for some of you to understand, in the sense that if you have a cell biology, you will understand those slides. If you don't have that, it's a little bit difficult. And I just didn't want to distinguish because cell biology is not a requirement for that. So I just wanted to pick up the topics that everybody can understand, whatever background you have. So that was the basis, not that one slide contain less information, more information, better. It's just like that. All right, so the first one in the special topic, this is from Kirsten. And what these scientists actually did, that they just looked at the pattern after infecting the cell. And this is all done by scanning electron microscopy. So basically, you infect in different MOI, 0.1 MOI, 2 MOI, not 2 MOI, they just 0.1 and 1. And then you just take the cell, fix them. Remember that these are not lung cells. These are the Vero cell line that they used, but the virus grows in there. One of the interesting things that they found, that the virus-infected cell increase the cell surface protrusions. And this is not unique. We have seen this many times because one of the reasons that the cells actually do that, remember that they present the viral peptides with the MHC1 on these projections, so it is easier for the NK cells to find them and kill them. So we see that. So that's okay. The thing that is unique, and this is the first time it was reported in here, and still it needs to be determined because it's very difficult to identify these phenomena. What they are showing in this paper, that on the left-hand, the bottom picture we are talking about. So right here is one cell, and here is another cell. And what they are finding, that they are finding these philopodia. This is the word that you underline. Is the philopodia. We have seen that on top of the philopodia, the pox virus actually surf on that. And this is the first time that they are showing that they have other evidences here, but these round structures that they are seeing, that these are the coronaviruses. And these philopodia, they contains actin inside, and there is a hollow tube, and this is basically called a nanotube. And this could be a mode of transmission from cell to cell. Now, why this is important? The importance is that if this is the mode of transmission through these nanotubes, this can hide from the immune system, basically immune surveillance. Most of the time, what happens? You have the cell infected, it just gets out by the exocytosis, it's floating around in the body's interstitial fluid, gets to the next cell. In that process, through the receptor-mediated endocytosis, binds to the ACE receptor, gets in. You know that whole thing. Here, you do not need any receptor. You do not, well, the thing is that that's a questionable thing, that whether you need a receptor or not. But basically what can happen is that if you remember that you have this virus, it does have an envelope, somehow this nanotube or the philopodia can push the virus into the plasma membrane. If the envelope of the virus and the plasma membrane of the next cell touch each other, without any receptor engagement, the cell they can get into, because the phospholipids and the phospholipids will immediately coalesce in between that, without that. So again, those are the bigger picture, but all you need from here is the surface protrusion on the infected cells help the spread particles through this surfing. And it was proposed for the first time that a virion involved in the surfing of the philopodia. Now, remember that this is an additional mode of transmission. This is not that there is still the mode of transmission is receptor-mediated endocytosis that we have seen. So this is an additional mode of transmission. All right. So what we can ask, a type of question, is that which of the following mechanism has been proposed other than receptor-mediated endocytosis for SARS-CoV-2 transmission from cell to cell? And it's very straightforward, is the philopodia-based surface protrusion is also the correct answer. Very straightforward, no complications. So everybody good? Yeah, so if you just remember the philopodia and spreading through that will be all right. All right, so after that, we are looking at the immunopathology with the interferon cytokine response. So here let's take a look, and then we will go into the whole mechanism, what is happening. So here what happens is that when the virus gets into the cell, I'm just looking at the figure on the right, the virus gets into the cell, and automatically the viral response is to produce this IFN alpha beta, which is the interferon 1. So lambda is a interferon like a class 3. That's not important. But basically what happens, we have seen by various mechanisms, the virus is going to block the IFN production. Now what happens, the virus replicates, gets out of the cell, and then where it finds that rather than initial checking of the viral replication, finally These are the immune cells that what you are seeing right here. The immune cells actually now finally get handoff over the virus. So what is the problem in between these two cases is that at this point, you do not have any interferon alpha, beta, or lambda at the initial stage. And suddenly, once the immune system see this, you will have a burst of IFN alpha, beta, lambda, also TNF, tumor necrosis factor alpha, and the IL-6, IL-1 beta. All of these names that you are seeing, these are all inflammatory cytokines. And the burst of this is going to cause what we call as the cytokine storm as well. So here you can see that after that, what happens? That you have an unchecked viral replication, and as you have this unchecked viral replication, so it starts from here, it goes like this. So this unchecked viral replication, also the monocyte, macrophage, dendritic cells, they are going to secrete the alpha, beta, lambda, and this will cause the cytokine storm as well as the inflammation. These two are the key in the immunopathology in that process. So the first thing is that what we are looking at, that IFN1, interferon 1 is your IFN alpha and IFN beta. Alpha, beta, these are class 1. And the release of the timeline determines the degree of immunopathology, but mainly this is the most important part, is that delayed IFN1, means both alpha, beta, response increase the disease severity. So see that it is blocking the alpha, beta. So delayed IFN class 1 induce the cytokine storm, increase the release of inflammatory cytokines which cause immunopathology, and some of the important ones are TNF alpha, IL-6, interleukin 6, and interleukin 1 beta. And then what happens, that the later part, what happened, there is also another, not later part, this is also another IFN class 3, IFN lambda. This, they also, the IFN lambda also induce antiviral state. But the function of one of these antiviral state is to suppress the cell division and differentiation. So this is one of the things, because remember that the cell wants, the virus wants the cell to divide so that they can replicate in those dividing cell. And the cell doesn't want to divide so that virus can stop the viral infection. So the lambda suppresses the cell division or the differentiation. Now see that where the problem is. The problem is that they release this TNF alpha, IL-6, Y, and beta, and these are so much in the process, they kill the pneumocyte, which are the lung cells. So the lung cells are being killed. Now what is happening, you have the lambda, these lung cells cannot replicate and replace the dead ones. So you have a sustained tissue injury. You do not recover from the tissue injury. Lungs is very resilient, but unfortunately, the lambda stops that resiliency. So this works against you. So that's what I wrote. Understand the rationale, the suppression of the cell division differentiation to restrict the viral replication. So the cells are killed by these, and then you have no renewal of these cells, but due to the lambda, that causes the problem. All right, so let's take a look at what kind of question that we can ask is that one of the important one that we can use that say, for example, say if I just ask is like which of the following is implicated in the SARS-CoV-2 induced cytokine storm. You just remember that for the cytokine storm is usually the delayed IFN class 1 induced cytokine storm. Just remember that the lambda is not, but again, just IFN alpha beta, delayed alpha beta response is basically cause the cytokine storm. And why it is not lambda? Because lambda induce a different set of inflammatory response. It doesn't cause the inflammatory response. Inflammatory response genes are mainly caused by the IFN1, which is the alpha beta, and it's a delayed response cause a burst of inflammatory cytokine secretion. Basically, what happens when the body gets the shock in a delayed, it just produces too much to fight, and that too much basically kills your cells. So again, so that's your important line. Delayed IFN class 1 induced cytokine storm. So IFN alpha beta, that's IFN 1, and that is the release timeline determines the degree of immunopathology. So just remember IFN 1, IFN alpha beta. That should do it. All right, so let's go to the next one. So here what we are looking at, we're going to look at several different types of SARS-CoV-2 protein, and these are coronavirus proteins. Some of them are not, some of them are. Like some of them are found in other coronaviruses, but this is just only one example of that. But most of these are common in all coronaviruses, and here is one protein, which is NSP1. This is at the very front of the whole genome, at the very front. The first gene that is in the polyprotein is the NSP1. And what it does is very interesting. It has an N-terminal domain, C-terminal domain that you can see. And the C-terminal domains actually binds to the bottom part, the blue part that you are seeing. This is the 40S ribosomal subunit, which forms the initiation complex, if you just remember from your translation in there. And when it binds in there, there is a tiny channel through which the mRNAs enter, and it scans for the initiation complex. It blocks that channel, and mRNAs cannot just enter in there inside the initiation complex. No pre-initiation complex is basically formed. And this is how it blocks the host mRNA translation, basically. So that's the thing, the host translational suppression. So the mRNAs cannot enter in here. Now what happens, that this is the most important part, and this is already well known. All of the viral mRNAs that are produced, they have an extensive secondary structure in their 5' UTR, untranslated region. And this 5' UTR binds to the N-terminal domain of the NSP1. So what happens when these RNAs are coming for scanning, any available NSP1 binds to this UTR right here, and there is absolutely no problem because this basically titrates out the NSP1. There is not enough to bind and block the RNA channel, and the viral RNAs are perfectly fine, and they can be translated. So basically, this is the difference in between that the 5' UTR secondary structure basically titrate out the NSP1. So the viral mRNAs are translated preferentially, whereas the host translation is totally shut down. So this is a very, very unique and one of the very well-established virulence genes that is basically the mechanism. So here we will ask, the question is the mechanism. That means that we will ask like this, is that how NSP1 inhibit the host mRNA translation. So your answer will be, it binds to this 40S ribosomal subunit and blocks host mRNA blocks the host mRNA for the translation. Again, all those channels and all those not needed. As soon as you see that we just give you an option of binding to this 40S ribosomal subunit, which is the smaller subunit, and blocks the mRNA entry or blocks the initiation complex formation, that will be basically the answer for that. All right, so very straightforward, no complicated, nothing complicated. So let's take a look at the next one. This is by Rachel. So here, another protein, and this is NSP14. And this is another protein, NSP14, and it has an entirely different function. So the part of the NSP14 is exonuclease, so it does the proofreading. The C-terminal domain of that is a methyltransferase, and it works in association of this NSP10. So basically, NSP10 increases the processivity of the NSP14. So this is the primary function of the protein. So basically what happens is that NSP10, without that, They form the complex, and one of the proofreading exoin activity that can do that, exoin, and also adds the methylation at the N7 MTase, N7 methylation is also done. But what happens that now this paper, which was a PNAS paper in 2021, is the first time it has been implicated that NSP14 is basically doing a global host translational shutdown. And I could have put a star marks in there. It was like, this is as easy as the previous one. Why you have not star marked or put a question in there? The difference is that in the previous one with NSP1, we know the mechanism, that how it does that by binding to 5' UTR. Here, the PNAS paper reports that this happens. We do not know that how it happens or how it does that. They speculate that it may be still the host mRNAs, or somehow it blocks the host mRNAs to enter the ribosome, and maybe that the same mechanism that the viral RNAs with the 5' UTR are preferentially translated. But the mechanism is still not known. This is the first report that they're saying that the NSP14 is a potent global translational inhibitor. That means that it just blocks all host mRNA translation, but doesn't affect viral mRNA translation. But the mechanism is still not known. And another thing that they are reporting, that NSP14 is probably even more potent than NSP1. They compared those two. So that's basically what it is. So NSP14 is the second SARS-CoV-2 protein. So very interesting that how you can have this dual functionality of these proteins. It's like really amazing. Think about this way, that if you just think that, okay, all right, the protein is translated by the ribosome in the cytoplasm, and the function exo-N and MTase, this is related to the replication transcription of the virus. So this is transported into those double membrane vesicles in which forms the replication transcription complexes. So a batch of protein goes in there. Other proteins stay in the cytoplasm to block. So two different distribution and extremely complicated process overall. All right. So the next one is from Bree, and this is basically the SARS-CoV-2 antibody cross-reactivity with other human coronaviruses and the duration of this. And this is a very interesting slide in the sense, and we are still collecting data. So I'm going to say that. So what we are looking at, that the blue ones on the top picture, the blue ones, 229E, NL63, HQ1, and OC43, all of these are actually the human coronaviruses. These cause 15% of the common cold. These are endemic in the population. Once in a while, you are exposed all the time to these coronaviruses. Everybody are exposed. Think about what happens. So basically what happens for most people, once in a while, we got a cold, which may be caused by these coronaviruses. So we will have an antibody against that, any of the strain. But what happens that in future also sometimes we are getting exposed to these viruses, and automatically that exposure will act as a booster, basically. So due to that antibody level, you may have a asymptomatic exposure, which keeps your antibody level high, or you may have a symptomatic cold. After that, you will have a higher level of antibody. So the constant exposure to these different strains, see that what happens, this is the level or seroprevalence. That means that in the serum, how much of these antibody level you have. Right here, it is a steady level. That's your seroprevalence, basically, antibody levels in there. But for the SARS-CoV-2, this is like, again, just think about that. This is a very short period of data. And what we are seeing, that approximately about 6 to 8 months, the level of this is going to decline. Remember that, the rationale of that. if the SARS-CoV-2 or the MERS or the SARS-CoV, in any of those blue ones, those are the ones which are the pandemic strains. People do not get exposed to them in as frequently. You are probably going to get it for one time, and then your antibody level is probably going to decline. So that's the normal. And can we tell that whether if you just get the natural infection, whether this is going to protect you for the long term, you're not going to get a second one, or if you are having the vaccine, after a booster or two, you do not need any time. Can we predict that? No. So we have a Lancet paper that we saw that, and this response varies completely at the individual level. You cannot still have a data. For some people, even after 6-8 months of the study, the antibody level is good enough that they will be able to neutralize the virus. They will not going to probably get an infection. For some people, it is as close as 4 months, it's started dropping. So these are some of the problems that just try to understand the difference in between that. If you have endemic strains, once in a while you are constantly exposed. That's why your seroprevalence is much more. And this gives you the immunity to some extent to these, but this is the problem with that. And here is an interesting part. So this last box that we have, that we made. So this is not there. So this green box, green was not there. This gray and yellow was not there. It was all blank because there was no data. So when the paper was published, so we got some supplementary paper and then filled this one up in there. So this red box was not in this original paper from where it was. So if you just look at that, of course, what they're looking at, that the cross-reactivity between the strains. So of course, if you have a 229E, 229E, of course you have a cross-reactivity. So if you have a SARS-CoV-2 and SARS-CoV-2, of course you have a strong immune response if you have that. But what is that if you have any of these viruses prior to this, what is the data? So far the data shows, see that I just put a little bit of yellowish at the top and the rest is the gray. So the yellowish weak response to no response. So what does that mean? That means that it has now been shown that how some of the antibodies of your human coronaviruses, those blue strains, 229E, NL63, HQ1, OC43, they do cross-react with some of the epitopes of the SARS-CoV-2. But that may be implicated that some people are not getting the disease. May be implicated. This is not known. But for some, maybe they do not have enough amount of endemic HCOV antibody, and that may cause them for a symptomatic infection. So this is one of the gray area. It's not easy to determine this quick, because this data will take like a 5, 6, 10 years to understand at the population level. So that's basically what it is. It is not that the human coronavirus antibodies do not cross-react with these, but usually they are the weak response. Because on the other hand, you can see that right here, the OC43, right here, human coronavirus OC43, and the original SARS, you get a strong response, heterologous. That's pretty good. On the other hand, and SARS-CoV and the MERS, which is a Middle Eastern coronavirus, that's also get a pretty strong response right here. So they do cross-react. So will over a period of time this will change? I mean, like the thing is that all of these is probably going to be a factor in the pandemic, that some of the people are naturally, to some extent, will be immune to the SARS-CoV-2. Some people are getting the vaccine. Some people got the natural infection. And all of these basically probably will lead to the herd immunity collectively. And hopefully we will have a higher level of the SARS-CoV-2 seroprevalence. And that's probably going to contain the disease. So very interesting paper. All right. So after that, again, another slide, which is important here. which is the suppression host defense by the SARS-CoV-2. And we are talking about two new different proteins in here. So let's go one at a time. That what happens, that here the protein that we are talking about is NSP16. And see that this also have an entirely different function. This is a 2' O-methyltransferase. The other one was like when we talked about the NSP14, we saw that that was a N7 methyltransferase. So in N7 position, it adds a methyl group. Here it's a 2' O-methyltransferase. It puts it in a different place. We have seen that in the previous section, module 2. But see that this is a dual function. So again, as we can see, that as a methyltransferase, after the protein is produced, this should go to the replication transcription complex in the double membrane vesicle. But instead of that also, this is transported, the protein is transported inside the nucleus of the cell. So it has a nuclear import signal in that protein. I mean, like that is like how the protein, these viral protein can be dual function. And see that what happens, that the 2' NSP also translocate to the nucleus and bind to the E1, E2 snRNAs. So these E1, E2 small nuclear RNAs, these are the integral part of the spliceosome. When NSP14 binds to these RNAs, basically the spliceosome, the host mRNAs are not spliced, cannot be transported out of the nucleus. I mean, like, see that how intricate these mechanisms are. And there is basically no mature mRNA because there is no splicing. Without the splicing, it cannot be transported. So here we can just ask some question like a critical thinking question. So say for an example, if we just give you an example like that. Suppose you are having a cell culture and you have infected your cells in the culture with the SARS-CoV-2. And then you are looking at the cells and you somehow found that a large number of RNA accumulation inside the nucleus. Again, I'm repeating. The infected cells have a large amount of RNA accumulation, mRNA of course, inside the nucleus. And what will be your implication? Does that make sense? So basically what is happening that the NSP16 binding to the spliceosome, E1, E2 small nuclear RNAs, and the splicing of the mRNAs that are produced, the primary mRNAs are not happening. They're stuck inside the nucleus. No splicing, no export from the nucleus. So that will be your rationale that why there are some stuck host mRNAs inside the nucleus. That's what it is. The viral, it has nothing to do with the viral mRNAs. They are produced in the RDCs, and there's no problem with that. So here is the NSP1 as well. That's the same thing that we talked about. That's NSP1. This was the previous slide. The same thing that we. This was the same slide, NSP1, that it just blocks. There is no mRNA scanning and there is no initiation complex formation, and that's what it is. The other one is also that, see, NSP8 and NSP9, these are also interesting, and these NSP8 is an RNA polymerase processivity factor. So NSP7 and 8 basically produce a pCNA like a clamp-like structure for the RNA-dependent RNA polymerase. That detail is not required. And NSP9, on the other hand, is a single-strand RNA-binding protein. That is also the replicase complex. So the bottom line is your NSP7, your NSP8 as NSP9, they are all in the RNA-dependent RNA polymerase holoenzyme as the subunit and support the transcription and the replication. And here is one of the interesting way that it binds. The NSP8 and 9 binds to the signal recognition particle, SRP, and disrupt the secretory membrane or soluble protein synthesis in the ER. So see that I'm not going to ask a question out of that. There is no yellowish line in there, because if you have taken cell biology, you completely understand this. If you have not, then it is a little bit difficult. So just in a nutshell, still I'm just telling you that what it is doing and what SRP is. So the ribosome starts, for the host protein synthesis, the ribosome starts synthesizing the protein in the cytoplasm. As soon as there is a signal that comes out from, which is in the form of a signal peptide or internal peptide, comes out as a protein signature out of the ribosome, the SRP, or the signal recognition particle, immediately goes and bind to that. Usually they're hydrophobic, bind to that. And the SRP, along with the ribosome and the mRNA, bring that whole particle onto a SRP receptor on the endoplasmic reticulum. So it was a free ribosome, mRNA, protein. It is translocated by the signal recognition particle on the ER. Now from the ER, it transfers to a translocon, which helps the protein, either if it is soluble, to put inside the ER lumen, or if it is a membrane protein, it transfers to the translocon on the membrane of the ER, and then it follows the secretory pathway. So from the ER, it will go to the Golgi. From the Golgi, it will be transported either to the plasma membrane or lysosome, or secreted outside the cell. So that's the secretory pathway. So SRP is essential for the protein to come onto the surface of the ER. If it is not going in there, there is no secretory protein synthesis. So that's basically a large, there are 1,500 just only plasma membrane proteins. Think about that. The second part is that all your interferons that are produced, the interferons are secreted out of the cell. If you block the secretory pathway, interferons are not going to get synthesized, transported outside of the cell. That's basically no antiviral response, and that is basically the defense mechanism. So you can see that there are so many of them, NSP16, NSP1, NSP8 and 9, also NSP14, so many of them. But here, this is the most important part. Just remember that the mechanism, how NSP16 blocks the mRNA export. So if you see, basically, again, I'm repeating, if you visualize in a scenario, there is a large accumulation of the host mRNA inside the nucleus, you know that this is basically the mechanism. NSP16 is binding to the U1, U2 spliceosome, small nuclear RNA, inhibiting splicing, thus inhibiting export out of the nucleus. All right, so let's go to the next one. So here, this is a very interesting paper as well, is that many scientists, they were all trying to do many different cell culture work with the SARS-CoV-2. But what happens that it is always difficult to have a proper model. Say, for example, if you're doing it in an animal, it's not human. If you are doing in a cell culture, it doesn't mimic the exact alveolar condition or the cell. So what they did, that they actually first did a 3D cell culture alveolar model and look at the viral replication transcription in that. So this was basically the main thing that what happens. And when they have these, that they looked at the viral titer goes up 1 day post-infection, and the expression to cell adhesion molecule, basically this is downregulated, and basically likely caused by virus to inhibit recognition by the immune cells, and also the expression of the interferon stimulated genes basically goes up, and likely caused by the HAT to the induced immune response. So here is one of the interesting part that what you can see, that in the interferon response is high in the modest viral burden. So what happens that when you have a modest viral infection, remember that you are not getting not likely to get a cytokine storm, because and why it is so? Because there is not, so this will, you can connect the two dots, that less viral load means less of those antiviral, less of those virulent protein, NSP1, NSP14, 8, 9, 16, there will be less. So some of the interferon can be secreted, and as some of the interferon could be secreted, your immune system is primed in a proper way. But if you have a high viral load, unfortunately, what happens in that case, your high viral burden, you have a delayed immune response, and this finally, when the immune cells actually see the type 1-2 interferon by the immune cells, you have an overburden of that. So this is interesting in the sense that the model, rather than just doing the cell culture, a 3D alveolar model culture can mimic the lungs much more in a much more defined way, and that was basically what was observed. But most of the time, they are just looking at most of the cells are having a single viral entry, basically. All right. So after that, this paper actually talks about that how the SARS-CoV-2 escape from the cell autophagy. And this is also, and I did not go into the detail of this or did not highlight, because you will understand this if you have a cell biology. If you not understand this, or it will take a little bit more difficult you have to read the narrative completely to understand that. So let's take a look and just explain that what is happening here. So in a normal cell, you have these autophagosomes, and these autophagosomes, just think about that this is an endosome, and double membrane endosome, and then what happens, by the help of these hops, which is associated with this lysosome right here, this autophagosome is going to get transported towards the lysosome, and they are going to meet each other and then finally fuse. So here you are looking at some snare. So what are these snares? So in the process of transport, there are several different things goes on. First of all, on the surface of these autophagosomes or the transport vesicle, there are some proteins which are the GTPase, which are called the RAB. There are 150 of them. So the RABs do have a cognate receptor on the destination. So you have a RAB here that understands the destination it first binds. Along with that, there are some other proteins which are called the snare, and there are two types of the snare proteins. One is basically the V snares, the vesicle snares, and then T snares, which is basically along with its destination. So these V and the T snares basically interact, and if you remember that you saw that when the viral membrane fusion happens, those fusion peptides falls in, and there is a hairpinning, and the two membranes are brought together. You remember that, those animations, that how that they just roll up and just bring the membrane together? V and the T snares actually do that. They will form this alpha helices bundle and pull those membrane, which you have. So these together will bundle up and pull these two autophagosome membranes with the lysosome so that they can fuse. So that's the overall view of that. So what happens is that the ORF3A protein, and this interacts with the VSP39, and this is a part of the Hops complex. And this is basically prevents the fusion of that. So what is the function of that? VSP39 basically interacts with the RAB7, which prevents the interaction of RAB7 to the VAB7-8 and SNAP29 in the snare complex. So basically, let's explain that what happened. RAB7 is on the autophagosome, and Hops is basically understands the RAB7. When RAB7 and Hops interact, the T and the T snares will come together and roll up. VAMP7-8, SNAP29, these are the snare complex proteins. So right here. And the interaction prevents the lysosomal tethering, bringing the autophagosome lysosome together and the fusion of the membrane. So basically what happens, the C-terminal and the TM transmembrane domain require for the autophagy to block, and that's basically the function for the ORF3A. So the ORF3A blocks, and the RAB7 cannot recognize the hops, and there is no fusion with the lysosome of this autophagosome. So so basically what happens is that you have a receptor-mediated endocytosis, And that by the time the TLRs in the endophagocytes understands that there's a viral infection, so they want this autophagosome to fuse with the lysosome to destroy the virus, but the ORF3a blocks that process. That's your global message. All right, so right here you have an imbalanced host response, and we are talking about this many times. That again, remember that this is also a recap, that patients show low levels of IFN1 and IFN3 and a high chemokine signature in the immunopathology. And IFN1-3 play antiviral and anti-proliferative immune response. As you can see, that anti-proliferative immune response means that it actually checks the cell division that we talked about. IFN lambda, which is an IFN3 that blocks the cell division to check the viral replication. And the low level of the cytokines cause an improper recruitment of the inflammatory monocyte macrophage population and the development of immunopathology. And then what happens? So the IL-6 and IL-1RA are elevated in the patients. Now, what happens is that IL-1RA is also an inhibitor of a pro-inflammatory cytokine, which is the IL-1 beta. But even the IL-1RA cannot, when the IL-1RA is elevated, IL-1 beta basically should be suppressed. But what happens, the cell wants the inflammatory response due to the high viral load. IL-1 beta is elevated. The RA is trying to inhibit that. It just loses the battle. But that high level of that, as it is being inhibited, its level, the cell tries to make it more of the IL-1 beta, and that causes the inflammation. If you guys go back to the slide, remember that the inflammatory cytokines, like you have a TNF alpha, of course IL-6 as well as IL-1 beta is also implicated in that. And this corresponds to the cytokine release syndrome. High level of cytokine may increase the inflammation throughout the body and interfere with the multiple body function. So here is the rationale that I talked about, that if high level of RA tries to inhibit IL-1 beta, the cell tries to produce more along with the other inflammatory cytokines. So that's what the problem of the delayed response is. All right, so these are some of the inflammation, but we did not talk about the CXCLs. CXCLs are also inflammatory cytokines. We talked about the IL-6, IL-1, and these are IFN beta, lambda. We talked about the IFN alpha as well, which is the IFN1. All right, so these are the conditions. The next one is what we are looking at. Let me see this one right here. Yeah. So the next one is basically the trafficking, because remember that our first chapter was basically assembly and trafficking. And although it just shows the full thing, we are not looking at the full thing. We are just looking at this nucleocapsid, spike, membrane, and envelope protein synthesis and the assembly. Just the left part we are looking at. So basically what happens that all of these proteins, once they are produced, they are recruited along with these ER. And these ER, so you can see that this is the envelope protein, then you have the membrane proteins right here, the blue and the green ones in there. And then the RNA comes in, the genomic RNA, which is enwrapped with the nucleocapsid protein N. And along with that, you can see that how the right here is the spike protein. Of course, the S is the spike. So you have the envelope, S, envelope, and the spike, they are already in the membrane. And now you have what we call as an ER-GIC, ER, and here is the Golgi. And this is your endoplasmic reticulum Golgi intermediate complex. So this ER-GIC is where the viral assembles. So think about that. This is a phospholipid bilayer, and you have all the proteins in here. The green ones are the spikes, then you have the red ones are the envelope protein, and then you have the blue ones are the membrane protein. And here you have the nucleocapsid enwrapped genome RNA, plus strand genome RNA. this whole thing is going to enwrap this. Just to visualize this, that this whole thing will enwrap this, and now you have the virus with the membrane. So right here, what you are looking at, that the purple round structure that you are seeing, that's basically this portion of the phospholipid bilayer. So this envelope is basically derived from the ERGIC. This becomes the viral envelope. And this membrane will go, so this membrane is basically the outer membrane that you are seeing, which will fuse with the plasma membrane to release the virus out. So again, E and M modulates the N-glycosylation nucleocapsid, and the N-glycosylation is also required for the proper folding and the stability, because these needed, the membrane curvature is very important to enclose the nucleocapsid protein, and all of these structural proteins are required for optimal formation of the subviral particle. If you just block the synthesis of just one of them, you are not going to find. So for an example, if you stop the M protein in a recombinant virus, this has not been done in SARS-CoV-2, in the other coronaviruses. If you stop the membrane protein synthesis, you are not going to get any viral structure is there. All of them are so important in that process. So this is basically the assembly site. The endoplasmic reticulum Golgi intermediate complex, ERGIC, is the key thing. So that's basically the key out of this. All right. So here is also, this was probably one of the most complicated paper and Kat did this very good job of summarizing this all together. So what they did is that they studied some of these antibodies, and the antibodies were from two different sources. First, they raised these monoclonal antibodies with these mice, and these are called veloimmune mice, the VI mice. So these are commercially available. If you have not heard about these VI mice, these are literally Frankenstein mice. So what they are done in a transgenic in the sense is like an unbelievably transgenic. They have deleted in these mice every B cell and T cell genes in those mice. They do not have a single of the T cell or the B cell genes. They are all replaced by the human T and B cell genes. So think about that. It's whole, I mean, like this is the way that you can edit the genes now. So it's a mouse, but it's going to make these B cells and T cells like the humans. They have all the receptors are going to be the sequence of human receptors, basically. And they will perfectly function in a normal way because they will secrete the antibodies, react with the antibodies. They are in the mice, but they are basically expressing the human antibodies. And these are those are your humanized mice, basically. These are commercially available, but they are create a recombinant of fully human antibodies. So this is kind of like a very, very interesting. So you can easily look at these veloimmune mice as such, that what they are. Along with that, also what they did, that they also look at the antibodies from more than from the blood samples and collected blood and sorted the individual B cells. And then they found from these two about more than 200 neutralizable monoclonal antibodies they developed, and they did these cross-competition assays and picked up okay, all right, and picked up two of them which they think are the best. So these are your Regeneron 10933, and the other one is Regeneron 10987. So this work was done in the Regeneron, and what you are looking at right here, so space filling plus ribbon model, right here is the RBD domain of the spike protein. Remember that this is just only the receptor binding domain. This domain binds to the ACE receptor. It doesn't have the stalk. Remember that the whole protein will have a stalk at the bottom of it. You are only looking at the very top portion of the spike protein, not the stalk, because this is the most important part. This engages the ACE2 receptor for receptor-mediated endocytosis. The stalk is pretty much insignificant in that case. So they found two epitopes. So this is epitope 1, a small stretch of amino acid, and this is another stretch of amino acid. And these two they found are probably the two most potent monoclonal antibodies which bind into the receptor binding domain. And that was basically the work, and that went to the clinical trial. And this was the interesting part, that President Trump, when he just went to the, was taken to the hospital in the NIH, he received this, at that time, this was at the clinical trial. He received this cocktail, these two, and this is now FDA approved. But the problem is that, remember that from these B cells or the recombinant fully human antibodies that is produced in the mice, all these monoclonal antibodies, you have to make a hybridoma produced in cell culture and purify that, then inject it. It's an extremely expensive process. So the therapy will cost normal people like $30,000 and up. And I'm not sure that whether insurance cover it or not, but probably it is doing that. So in extreme cases, the monoclonal antibodies, when there is no other way, the monoclonal antibody shop is probably, without others, is probably the most effective ones. If there is, now remember that what the problem is also, that if you have a mutant strain where the epitope has changed, this monoclonal antibody is not going to work. That has been seen. So it has to be the one that is infecting. And there are many, many companies actually came up with many different sets of monoclonal antibodies. And in extreme cases, this is the best therapy that still we have. But what was the big news yesterday or day after before from Pfizer? Anyone? Yeah, they applied for, this is called, what is it, Lofizupit or something like that. So they actually applied for the FDA approval of the first drug against the SARS-CoV-2, and they are saying that it is 90% effective. So how it works is kind of interesting. It's not an entirely new drug, and it is basically a modification of a ritonavir, which is an HIV protease. So what they're saying that they modified this drug, and if you remember that we talked about the 3CL protease, which cuts the AB and A in small pieces. And if you just inhibit the viral 3CL protease, which is the main protease, you're not going to get any individual protein, and that's the drug is doing. So I think things are going pretty well. I mean, like, it's really interesting that probably we may have finally an antiviral as well. But it was an HIV drug, though. It's not an entirely new drug. It is a ritonavir, modified ritonavir, which is a protease inhibitor. So we're going to see that what's happening in there. All right, so this one is the last one. Yes, this one is the last slide. And of course, there is a star in there. And here is also another very interesting protein. And ORF9b is an accessory protein. So what happens is, like, if you just look at the genome of the SARS-CoV-2, you have the full genome, and N-terminal part, it produces this polyprotein 1A and 1AB. Those genes are pretty much constant and fixed. Those are basically the non-structural proteins, including the RNA-dependent RNA polymerase accessory proteins. So we talked about NSP1, NSP14, NSP7, 8, 16. All of these are produced from that. That is produced in all coronaviruses. Whereas this ORF9b is a SARS-CoV-2 specific protein. It is not expressed in other coronaviruses. And it has been shown that it does the transcriptional inhibition of the IFNs in a very unique way. And I just, again, there's a lot of different things going on, but I'm just going to show you just what the key thing is happening right here. So then I will just read through here. So the TOM70 is, TOM70 right here, is a membrane protein which is in the outer membrane of the mitochondria. It's a transmembrane protein. And what it does, its main function is transportation of proteins inside the mitochondria. But I'm not going into the detail. It's a translocon kind of function that it does. But along with that also, it can activate other proteins. And in that process, one of the important protein is an HSP90 right here, HSP90, and it's a heat shock protein, 90 kilodalton in size. And these are some of the very important chaperone proteins which are implicated in the activation of these transcriptional cascade pathways. So what it is basically doing, so the TOM70 needs to activate the HSP90 and see what it is doing. This is known to you. This is also, it is bound to the IKK, and when it activates the HSP90, it releases this I kappa beta and NF kappa B. From this, the I kappa B is going to get proteolyzed by the ubiquitination, not needed, but we have seen this before, and NF kappa B is going to translocate into the nucleus for the transcription. And here is also another one. This is called a tank binding, TBK1, a tank binding kinase, going to phosphorylate IRF3. Phosphorylated IRF3 is going to go inside the nucleus and also a transcription factor. So basic bottom line is that TOM70 needs to activate HSP90 to activate both of these transcriptional activation pathway. And ORF9b is a protein that binds to the TOM70 and disrupt this pathway. That's basically the bottom line. So let's see here what happens. First, ORF9b is an accessory protein associated with TOM70, an imported receptor on the outer membrane of the mitochondria. This is not, but again, this already, of course, they are going to complex showed hydrophobic interactions, but it just binds. That's the main thing. And normally, HSP90, chaperone protein, binds to TOM70, leading to TBK1 recruitment of HSP90, and then phosphorylates the INK3 and induces interferon alpha beta transcriptional activation. But basic bottom line is like this. When ORF9b is bound to TOM70, HSP binding is reduced. Fold is not required. HSP binding is reduced, and this inhibits the interferon transcription and part of the innate immune response. So just the take-home message is basically what we can ask is that how ORF9b repress the transcription of the IFNs. So again, we're repeating how ORF9b repress the transcription of IFNs. And just remember that by binding to TOM70, this is your mitochondrial protein, and then inactivation of the HSP90s and the downstream and blocking the downstream signaling. So just only two things here, just ORF9b plus TOM70, no HSP90 signaling. So HSP90 underline that, TOM79 underline that, and ORF9b, and transcriptional inhibition is the global thing. That's it. That will do it.